Everyone, today I'll be explaining how the graphs of competitive inhibitors and non-competitive inhibitors are based in this form. So first thing, we see that the blue line shows the non-inhibitor, means it is the whatever the graph was, the narrative reaction of a substrate with no inhibitor. Alright? Now, if you wonder what is a competitive inhibitor, you should first thing learn what is competitive inhibitor and what's a non-competitive inhibitor. A competitive inhibitor is, for example, if you have here an enzyme and this is its substrate, right? You know the substrate, it goes into the active site as they're complementary. But in the case of a competitive inhibitor, there will be another molecule called the competitive inhibitor molecule that also has the same, that's also complementary to the active site. So what happens here, just from its name, it is competitive. So the competitive inhibitor and the substrate, they basically they go into a race to who will go to the active site first. Now in this graph, for you to know why did we plot them in this shape, you need to understand that competitive inhibitor is a reversible reaction. How? Since you are here trying to overcome the power of a competitive inhibitor, you should do something to do it. You can, if it makes sense, you can increase the number of substrates. Basically, you're increasing the number of substrate to be so more substrate than the competitive inhibitor. Now if you think about it, if you increase the substrate number, if you increase the substrate concentration, the substrate will be the one going into the active site, as there are more concentration of them, so they will be the one going into the active site, right? And the inhibitors, as they were like constant, so if there was an increased number of substrates, so the substrate will be going rather than them. So that's the way you overcome the competitive inhibitor, by increasing the number of substrate. Now if you're looking at the graph, it shows exactly what I'm talking about. Now the non-inhibitor is this. We have here is the Vmax, and here it's 1/2 Vmax. And now there is a Km here. The Km is for this non-inhibitor. Alright, and now if we're talking about the competitive inhibitor graph, it goes like this, right? You have here, it's 1/2 Vmax, and now something very important, you can see it's here, Vmax is the same as the past graph, which is the non-inhibitor, and the Km goes forward. Why? Now for you to understand what's a Km, Km is the amount of substrate needed for 1/2 Vmax. As you can see, in the x-axis is the substrate, here is the Km, and it is with the y-axis with the rate of action, which is the 1/2 Vmax. This is the meaning of Km. And again, if you look at the x-axis, you can see this, it is the substrate. The Km is in this axis, meaning it, again, from that definition, it is the substrate needed for 1/2 Vmax. So Km is talking about the substrate. Now if I went back to here, we said that we increase the number of substrate to overcome the competitive inhibitor, right? Since we increase the substrate, if it makes sense, we will go into the right side of the graph, so it will make sense that it will be here. The Km goes forward, means that we increase the Km, we increase the substrate, that's why there was an increase in Km for non-competitive inhibitor, right? Now this explains why Km is in the other side, since we increase the substrate. And now if we're looking at the Vmax, you can notice that the Vmax and the 1/2 Vmax, they are the same. They're just like the non-inhibitor and the competitive inhibitor, they both are in the same point. Why? Because what is Vmax now? What is Vmax? Vmax is the maximum active site of the enzymes when they are like fully accumulated. So Vmax is talking about the enzymes, and as we said, Km is talking about the substrate. Vmax is again like when all the active sites of an enzyme are accumulated. So here, as I said, compared to the definition, Vmax, since it is based on the enzyme, and again, when I explained before about the competitive inhibitor, there was no change in the enzyme. We did not decrease the enzyme, did not increase the enzyme. There was no difference in the enzyme. So that's why the Vmax stays the same. Now, after we finished, we can now start talking about the non-competitive inhibitor. Again, this is the non-inhibitor graph compared to the non-competitive, which is the green one. Let's talk about the non-competitive. Non-competitive, from its name, again, if you have an, let's put it up, if you have an enzyme once again, alright, and here is the substrate that goes to the active site, but the calcium here is, let me just change that a bit since we're talking, okay, again, this is an enzyme and here's the active site and this is the substrate wanting to bind into the active site. Now the non-competitive inhibitor is something that does not look like the substrate and does not go to the active site. It goes to a site different than the active site. So basically it goes into another site that is not the active site, that is not this. It goes here. So if you think about it, if something went into the enzyme, it will, like, if think about it, okay, it will destroy the shape of the enzyme. What holds the enzyme together are hydrogen bonds or ionic bonds, right? And when the non-competitive inhibitor goes and attaches to the site of one of the sites of the enzyme, it will in fact break down the hydrogen and ionic bonds. Breaking down the enzyme, then it will affect the active site, and if the active site was affected, means it will no longer be complementary to the substrate. Then a problem will happen that the substrate will no longer attach to the active site of the enzyme, so the enzyme is now broken and something, you could say that. So no more substrate enzyme complex, and that will affect it a lot. And based on this, you can understand that, as I said, enzymes are being destroyed and stuff, right? So you can now have an idea that enzyme concentration decreases because of the non-competitive inhibitors. Let's think about the non-competitive inhibitors are just mean people that they want stuff for themselves and they destroy the enzyme. And that's basically killing it. That's why the enzyme substrate decreases. That's why the enzyme concentration decreases. Now we had an idea that the enzyme decreases. As I said before, when we're talking about enzymes, we talk about the Vmax, and we're talking about the Km, we're talking about the substrate. Alright? But in this case, we're talking about Vmax. In this case, we're talking about the enzymes, we're talking about the Vmax. Now, after we explained everything again, that was the no inhibitor. And we're talking about the non-competitive inhibitor, which is the green one. Great. Now, you can notice, let's make it a bit obvious, pink color. You can notice here, alright, that they go down to the same Km. Now think about it, why? Why the no inhibitor and the competitive inhibitor goes down to the same Km? Think about it. Yes, because we said here that only the enzyme was changed. Did we change anything in the substrate concentration? No. Again, I'm so sorry guys, I don't know what happened, but it's okay. There was a no inhibitor. This was the non-competitive inhibitor. As we said, the Vmax was for the enzymes, and the Km was talking about the substrate. Again, as if you wanna notice, you can notice that the Km of both, you can notice the Km of both of them goes down to the Km. Why? As we're saying that we said that there was no change in the concentration of substrate, so that's why the Km will stay the same. Alright, that's why the Km in the non-inhibitor and the non-competitive, they are still having the same Km. Now, what is different? You can notice that talking about the non-inhibitor, go down, go down, go down, you can see that here is, now first let's talk about the Vmax, then we're going to go back to the 1/2 Vmax. The Vmax of the non-inhibitor is here, and the Vmax of the non-competitive is here. So the Vmaxes are in different like positions. Vmax of the non-competitive inhibitor goes down. Why? Again, I said that the enzyme concentration decreases because the enzymes are getting destroyed by the non-competitive inhibitor. So it makes sense why the Vmax decreases, because the Vmax is depending on the enzyme. If the enzyme decreases, the Vmax decreases. That's why the Vmax of the non-competitive inhibitor decreases. So you can see, and the same thing will be for the 1/2 Vmaxes. Now, the 1/2 Vmax of the non-inhibitor was here, and the 1/2 Vmax of the non-competitive inhibitor was here. Again, it decreases because of the enzyme decrease. And the Km was the same for both of them because we did not change any substrate concentration. That's all. Thank you guys.